Uh, so we'll be going live. So yeah. Good morning, everyone. Welcome to the Flora Fauna Science Foundation's talk series. In this dynamic series, we hear from the brightest minds and thought leaders on a diverse range that span the realms of Fasal se Safal. Thank you for joining us, and let's begin this scientific exploration with open minds and boundless enthusiasm. Today, we have with us a very special guest who is also our esteemed member, the person behind first complete map genome of holy basil, and the one behind so many aromatic plant technologies, having more than 100 papers and much more. We welcome Dr. Ajit Shasany, Director, CSIR NBRI, to the forum. Welcome, sir. We have with us Professor S.P.S. Kanuja, Founder President of the body, and I request sir to please initiate today's session with his thoughts.
Thank you, Dr. Manika. Welcome, Ajit, Dr. A.K. Shasany, Director NBRI, and my own student, whom I have grilled a lot. Welcome, Dr. A.K. Singh, Professor Ashwini Srivastav, many others who have joined, including the Umang participants of the mission program of Flora Fauna Science Foundation. सभी को नमस्कार, आपका स्वागत और अभिनंदन इस फ्लोरा फोना साइंस फाउंडेशन के मंच पर, जहां हम बातचीत करते हैं महत्वपूर्ण विषयों पर। और जैसा कि आप जानते हैं कि हर वर्ष फ्लोरा फोना एक थीम पर काम करती है, जो एनुअल थीम होता है उस पर फिर हम एक्सपर्ट्स, विशेषज्ञ जो हैं, जो इस विषय से जुड़े हुए होते हैं, उनको बुलाते हैं और उनकी जो समझ और उनका जो अनुभव है, उनका जो ज्ञान है, उसको हम आप तक भी पहुंचाते हैं और अपने लिए भी उसको प्राप्त करते हैं। सो वी ट्राई on every second Saturday of the month to have an expert of the field under the theme, the annual theme that Flora Fauna Science Foundation has every year. As you know, we started long back. Flora Fauna Science Foundation started in 2015 with some founder members, and that has expanded into many members. They represent the society from all angles, from scientific sphere, from students and scholars, from entrepreneurs, from farmers, and also industry. And not only that, even those who represent society as activists, as journalists, as mass communication people, they also are member of it. So it's a unique platform. And usually we say that science must reach society. And here is an effort where science and society together are translating it. It is not only scientific minds or scientists by profession, but also the society components, which are more scientific than sometimes the scientists themselves in answering the basic questions. So friends, I welcome you all to this very important platform. And when we had started on annual themes, we started with Khet Se Sehat, where we tried to bring cultivation of health, like medicinal plants or drugs or plant molecules and so on, from the farmers' fields. And then we entered into Poshan Se Roshan, where we talked about the nutrition, empowerment of society through nutrition. And we entered into Krishi Se Kushi, where we wanted to bring happiness through agriculture. And then this year, we are into Fasal Se Safal. Fasal Se Safal means the crops, their harvests, how can they be successful? And today I am very happy to have Dr. Ajit Shasany, who actually would be talking about a subject which usually gets into myths of people. There are misunderstandings about biotechnology. जैव प्रौद्योगिकी के बारे में कुछ मिथ्या भी फैलाई जाती है कि ये पर्यावरण के लिए अच्छा नहीं है या ये घातक हो सकता है, और इस तरह से कई बातें कही जाती हैं। तो जब आज का विषय चुनना था, मैं डॉक्टर शासनी के साथ बैठकर इसकी बातचीत कर रहा था, तो हमने ये तय किया कि हम इस तरीके से बायोटेक्नोलॉजी या जैव प्रौद्योगिकी को सोसाइटी, जो हमारे समाज है, हमारे किसान हैं, हमारे उद्यमी हैं, उन तक उसकी जो समझ है वो पहुंचाने की कोशिश करेंगे कि ये तो बहुत ही उपयोगी एक विज्ञान का ट्रांसलेशन है, उसका अनुवाद है, जिसको हम बहुत अच्छे तरीके से उपयोग में ला सकते हैं. So it is not that biotechnology is something we have to be afraid of. अगर उस तरीके से हम सोचे तो चाहे न्यूक्लियर साइंस है तो चाहे कोई भी विज्ञान है, उसके पॉजिटिव और नेगेटिव दोनों ही बातें हो सकती हैं, लेकिन उपयोग पर निर्भर करता है कि हम कैसे करते हैं. तो आज का जो टॉपिक है वो बायोटेक्नोलॉजी फॉर बेटर क्रॉप्स है. बेटर क्रॉप्स का मतलब वी विल हैव द क्रॉप्स विच विल बी मोर एफिशिएंट एंड आल्सो वैल्यू एनहांसमेंट. यानी कि ना केवल फसल को उगाने तक की बात करें हम, बल्कि फसल उग जाने के बाद उसके जो उत्पाद हैं उसमें किस तरीके से मूल्य संवर्धन हो सकता है, वैल्यू एडिशन हो सकता है, ताकि जो एक आय है उस उद्यमी की, जो कृषि द्वारा उद्यमिता प्राप्त कर रहा है, बहुत अधिक हो सके. मैं डॉक्टर शासनी के साथ जुड़ा हुआ हूँ 1986 से शायद. और जब उनको मैं मोलिकुलर जेनेटिक्स पढ़ा रहा था, तब हम बात किया करते थे कि जीन से फंक्शन तक कैसे पहुंचा जाता है. यानी कि कोई भी जो हम व्यवहार देखते हैं पौधे में, वट एवर वी सी एज ए फंक्शन इन ए प्लांट is a consequence of the genes that it possesses, and these genes together, all the genes and the system carrying these genes is called genome. तो जीनोम से जीन्स तक जब हम बात करते हैं तो उसको फंक्शनल जीनोमिक्स भी कहते हैं. और जो मेडिसिनल और एरोमेटिक प्लांट्स की बात करते हैं तो वहाँ मेटाबोलोमिक्स, क्योंकि जो मोलिक्यूल्स हैं जिनकी वैल्यू होती है, ऑल दोज मोलिक्यूल्स विच आर मेडिसिनली इंपॉर्टेंट यूजली आर द कॉन्सिक्वेंस ऑफ सेकेंडरी मेटाबॉलिज्म, एंड सेकेंडरी मेटाबॉलिज्म इज गवर्न्ड बाय set of pathways, and pathways are governed by enzymes, and enzymes are governed by genes. So that is how we reach genes to functions. तो आज मैं आशा करता हूँ कि आज की जो व्याख्यान है, द टॉक टुडे, द लीड टॉक टुडे ऑन बायोटेक्नोलॉजी फॉर मासेस, for everyone, biotechnology as the most acceptable form of science which can change the scenario, save the planet, bring in everything that is eco-compatible would be discussed. Just to give a brief point on that, I would like to say that while we talk of gene to function and say application of biotechnology, it is not necessarily that it is gene cloning or genetic engineering only or gene manipulation only. It is also gene editing nowadays being talked about. Dr. Shasany would be talking about CRISPR technology, which is gene editing or genome editing only. But let me tell you that this goes back into history, into mutations. The mutations which have been happening in nature, when we do it in a side-directed manner, then it is called genome editing. And that is what we have learned. And therefore, nothing odd about it. It is very much existing in nature. And then genomics, as I said, all the genes together put on the system that is called chromosomes. We call it as genome. And that genome basically shows the variation from species to species and within species also in form of varieties or genotypes and so on, or we can say wild germplasm or genetic resources. So those things become very, very important. And therefore, to understand any organism or any cell or any biological system, biotechnology is highly essential. And therefore, for applying it towards the advantage of entrepreneurs, वो किसानों के फायदे के लिए हो, वो उद्यमियों के फायदे के लिए हो, उसमें मूल्य संवर्धन की संभावनाएं हो, तो उसमें कुछ एक स्ट्रेटेजिक वे है जिसमें हमें जाना होगा, जो कि आज उदाहरण के साथ डॉक्टर शासनी आपको बताएंगे. और उसमें सबसे पहला आता है रिड्यूस द इनपुट कॉस्ट ऑफ एग्रीकल्चर. जो हमारी लागत आती है फसल को उगाने की, उसकी लागत को कम किया जा सके. इसका मतलब हुआ चाहे वो बायो फर्टिलाइजर्स हों, बायो मैन्योर्स हों, चाहे हम जो इंसेक्टिसाइड या फंजीसाइड्स यूज करते हैं उसकी जगह बायो प्रोटेक्शन एजेंट्स हों, इस तरीके की चीजें, और फिर उस जमीन को, उस मृदा को हम अच्छा कर सकें, वो सारी चीजें। फिर उसके बाद आता है क्रॉप प्रोडक्शन और प्रोडक्टिविटी, उसको कैसे बढ़ाया जाए, यानी कि ईल्ड जो है, जो फसल आएगी, जो हार्वेस्ट आएगी, वो कैसे ज्यादा हो सकती है। और फिर उसके बाद आती है पोस्ट हार्वेस्ट टेक्नोलॉजी, जिसमें हम उससे उत्पाद पैदा कर सकते हैं, उसकी प्रोसेसिंग कर सकते हैं, और उस प्रोसेसिंग द्वारा आप अगर एक वैल्यू मिलती है आपको एग्रीकल्चर की हार्वेस्ट से एक्स, तो आप 10x या 20x तक जा सकते हैं, तो 20 गुना आपकी संवर्धन हो सकती है उस मूल्य में। और फिर उसके अलावा स्टोरेज है, क्योंकि स्टोरेज में बहुत ज़्यादा स्पॉयलेज होती है, तो कैसे हम शेल्फ लाइफ इंक्रीज कर सकते हैं ताकि टमाटर सड़े ना, टमाटर अच्छा रहे। और ऐसा एक कार्य एनबीआरआई द्वारा किया भी गया है। और फिर बायोडायवर्सिटी को किस तरीके से हम उपयोग में ला सकते हैं। बायोडायवर्सिटी का मतलब, जब हम एक जो डोमेस्टिकेटेड प्लांट होता है, जिसको हम क्रॉप कहते हैं, उसके जो वाइल्ड रिलेटिव्स होते हैं, जो जंगलों में या अपने प्रकृति में पाए जाते हैं, उनमें कौन से ऐसे जीन्स हैं जिससे हम सीख कर जीनोम एडिटिंग अपने ही फसल में कर सकें। ये भी एक तरीका हो सकता है, और इसको सिंटनी किया जाता है, कहा जाता है। डॉक्टर शासनी का अपना जो काम ओसिमम पर है, उसमें जो सिंटनी ओसिमम के 8 स्पीसीज में देखने को मिली, वो एक बड़ा ही महत्वपूर्ण कदम बनता है कि हम बाकी फसलों पर भी उसका उपयोग कर सकते हैं। तो आज मैं आशा करता हूं कि आप सभी लाभान्वित होंगे, चाहे आप विद्यार्थी हैं, रिसर्च स्कॉलर्स हैं, whether you are a student, research scholar, scientist, or you are a farmer or entrepreneur, you all are going to get the advantage of understanding how biotechnology is going to change your life if you adopt it in the proper way through the proper research that has been carried out in research organizations. And that is where the role of Flora Fauna Science Foundation as a bridge between research organizations and the utilizers of that research comes, and we would always be keen to do that. So without taking much more time, although I have taken quite a lot of time, I'll like to request Dr. Neeraj Jain to formally introduce Ajit, and let people know that the speaker of today carries an experience as well as the knowledge that you would be interested in. Dr. Neeraj.
Thank you so much, sir. And it's definitely a great opportunity for me today. I have known sir as my PhD guide. I had the privilege of working with him, and it's just a great honor for me today to present his introduction. I'm a little nervous also today. So Dr. Ajit Kumar Shasany, who is the lead speaker for us today, he is fellow of reputed National Academy of Sciences and also National Academy of Agriculture Sciences. He is presently director CSIR National Botanical Research Institute at Lucknow. Before that, he was director ICAR National Institute for Plant Biotechnology, New Delhi. And prior to that, he was serving as chief scientist, Biotechnology Division at CSIR Central Institute of Medicinal and Aromatic Plants, commonly known as CINA. He has 146 publications to his credit, out of which 104 publications are international publications, and has a huge citation index of 5,437, which with an H index of 38 and a cumulative impact factor of more than 400. He has granted patents of total 101 to his credit, with 51 US patents, and he has 46 variety development to his credits. He has been guiding PhD students, out of which 17 have been awarded and 4 are still continuing with him as a PhD guide. He has also guided 62 MSc students. He has been involved in 30 projects, out of which 16 he has led as principal investigator or co-principal investigator. His research interests include plant diversity, metabolic engineering in medicinal and aromatic plants, and bioprospection for human health. New genes for phenylpropanoid, terpenoid pathways for aroma, plant immunity for crop production, to name a few. He has complete genome and transcriptome sequence of foliaceae to his credit, and this is the first genome, medicinal plant genome, and first genome to be done completely in India. Being a graduate in agriculture, he is also engaged in dissemination of technology to farmers for income enhancement and livelihood upliftment. He has been leading in the development of 39 clusters of aromatic plants in 22 districts of Odisha under Aroma Mission, and has been developing startup entrepreneurs. He has several awards and recognitions to his credit. As I already told, he is the fellow of National Academy of Sciences, Allahabad, National Academy of Agricultural Sciences, New Delhi. He has been awarded the Samanta Chandrasekhar Award in 2012. He has been awarded the CSIR Technology Prize for Biological Sciences and Technology in 1999, CSIR Technology Award in 2012 in Biological Sciences, CSIR Technology Award in 2018, Certificate of Merit in Biological Sciences on OSIMA. He has been awarded Professor Umakant Sinha Memorial Award, Indian Science Congress Association in 2003, recognition for Indian Drug Manufacturers Association for Best Patent in 2004, SOM Award for developing high-yielding Mentha arvensis post-C by Essential Oil Association of India in 2005, recognition for 50 Award 2005 for Rural Development to CSIR CMAP, recognition for Nina Saxena Technology Award by IIT Kharagpur in 2007. You, sir, and sir has been very modest to give a very brief introduction. Yes, over to you, sir.
The next, I should take over or what?
Yes, sir, please, please, sir.
Okay.
Yes, sir.
Okay, very
So,
good morning to everyone.
So yes, sir, you
Monica?
can start, please. Yes, sir, please.
Respected Dr. Khanuja, my teacher, actually, whatever biodata you have read, so many of the things I am fortunate enough to share with him. And also, he is the initiator of my career. I can tell you that when he told me to join his group in CIMAP at my formative stage, after passing out from IRI as a PhD scholar, then joining in ICR initially, Kasargod. So I came to Kasargod and joined his group, and that's the formative stage of my career. And I started working with him, and I don't know what which direction I am going, and I was going. So it all led to this place, wherever I am. So it's the blessings of teachers. It always matters. Having said so, also I am fortunate to provide me the opportunity in a crucial day. Tomorrow is Sir's birthday probably. So advance happy birthday to Sir. So this particular day, August 12th and 13th. So this is the coinciding second Saturday. So I am speaking. Maybe we can dedicate this thing to the birthdays of Sir tomorrow. So very happy birthday, sir. Advance birthday again, sir, to you. Also,
Ajit, this is also 25th talk. So this is a silver talk that you are giving for Flora Fauna Science Foundation.
sir, thank you, sir. I got this opportunity again. Earlier also, sir told me, but always you feel nervous to speak whatever you know, and from in front of the person from whom you have learned. So always it is nervous to face the crowd whom you know. Also the teacher speaking in front of the teachers. So for that reason, so let me confess, I have bonked once, and second time I cannot, and I will not, and I'm trying to speak whatever I know, and if something goes wrong, then you are there to correct me. So with this, I will start the presentation. Whether I don't know whether I have done the justice or not, but whatever you have told, sir, I will just give a run over those and give a glance of those things. I'll be sharing my slide.
So Alt and Tab.
Alt and tab.
Tab.
Okay.
So first of all, share the screen and then Alt Tab and you can go to the screen.
Alt and tab. I have just gone for that.
So
Okay.
start sharing and then you can go to the slide.
Okay. sharing.
Yeah.
Then
Now
alt and tab.
alt and tab together. Alt and tab.
Alt and tab together. Then gone for that.
Now on full screen tab.
It is okay, sir. Now.
No, so you haven't started sharing. So
First
you have to share the
come
screen,
back to Zoom.
Zoom, Alt tab, Zoom window.
Alt tab from Alt tab. Yeah. Come to Zoom.
Zoom. Then
Now
I'll
share screen from the bottom green one you have.
okay. Alt tab again. Then
Yes.
I have the screen. Okay.
Now you press it.
So you have the Zoom screen in front of you, right?
Yes, John print share I will tell, na?
A share screen. Yes, share screen, share entire screen, or share the window.
Enter screen. Alt tab. Then go on for this one. Then this one. It has come.
Actually, the screen sharing has not started.
No problem. You go back to share screen.
Mhm.
Did you go?
Sir, again alt
Yeah.
tab.
Come to Zoom. Press
Uh,
share screen without alt tab. Okay. Without alt tab. Or after alt tab, you come to Zoom and leave both the.
now I will go for share screen, sir.
Yeah. Share screen.
Share screen. Then it has come share cancel
Yes. Now
window
you come to alt tab again.
alt tab.
And now go to that full screen presentation.
Okay.
Press it.
It has come.
Oh,
No, sir. So you have to click the full screen mode here. Share, you have to press the share option.
from alt tab, when you press the full screen thing, then say share.
Share is not coming actually.
सर आप Zoom पर जाइए.
Zoom पर
Zoom
गया.
पे जाके शेयर स्क्रीन में नीचे एक ब्लू कलर का बटन शेयर स्क्रीन का शेयर का दिख रहा होगा.
ठीक है.
हाँ, उस को शेयर करना पड़ेगा. शेयर को प्रेस
क्लिक करो उसको, शेयर स्क्रीन
उसको क्लिक किया.
क्लिक किया.
उसमें एक फिर टेबल आ रहा है. शेयर टैब ऑटो विंडोज एडिट एंटर स्क्रीन इट इज कमिंग लाइक दैट.
यस सर. तो स्क्रीन में क्लिक करके शेयर का बटन प्रेस करना होगा. दिस ब्लू बटन नीचे.
ऑल्टर टैब शेयर अल्टर टैब आ जाएगा. अभी अभी अल्टर टैब करूं या नहीं?
हाँ, ऑल्ट टैब करो और उसमें उस पे छोड़ दो, उसको टैब को छोड़ दो, ऑल्ट वाले को पकड़े रखो और उससे माउस से क्लिक करो जो फुल स्क्रीन में.
नहीं आया. What I will do, sir, I will go back. I will just I'll do my own way.
Okay.
It is not coming.
Yeah.
Do that.
Yeah.
एक बार हो गया था शेयर.
Yeah.
तो आप ठीक है. आपकी स्क्रीन आ रही है. अब यू कैन गो टू योर पीपीटी.
अभी स्क्रीन आ रही है.
हाँ. नाउ यू कैन ओपन योर पीपीटी.
कैसे?
बस पीपीटी को. हाँ, राइट. वी कैन सी योर पीपीटी नाउ. यस सर. यस सर.
Then it will it has.
Yes sir, it's fine. It's totally fine. We can see your PPT in full screen mode.
Okay. So thank you again for giving me this opportunity to speak before you. So this is what the topic given to me is biotechnology for better crop and value enhancement in agriculture. This is the comparison. You have seen these slides many times. Dr. A.K. Singh might have also given you these slides. But this tells the story. So when in 1950, we have net sown area was 131 million hectare. Now 140 million hectare. Sir, what I will do, I'm not going to tell anything about ARMA mission or any other missions, the comfortable zone of mine. I'll come to the topic biotechnology for food and all these things. and whatever you have told. So I will touch upon all the subjects like this because I was in also ICR and IPB. So I think I will be speaking on those things. Then fruits and vegetable production is 25 million ton in 1950, 330 million ton right now. 51 million ton food grain production, 314 million ton. Like this, if you see all the sugarcane, cotton, spices, oil seeds, it has increased a lot. But 131 million hectare, earlier it was there, now also 140 million hectare. Marginal increase. We cannot say this is the increase as such, but tremendous increase in production. So what has happened when 1950 till 2023 or 22, keeping the net soil area the same. So there is huge increase in the productivity, huge increase in the productivity. Simultaneously, from 1950 to right now, if you see the subpopulation, it has increased a lot, again, many times, many fold, and we are increasing. So the net sown area, productivity has increased, and this productivity increase, how it has increased? Because of the green revolution, one we can say like that. So when we consider the green revolution and aftermath of that, so we have given a lots of fertilizer, chemicals. Also, we have our breeders. They have only pointed the characters for high yield, and high yield. So we only concentrated on high yield. Also disease resistance, pest resistance to some extent. But at that time, our population of India needed food, and PL 480, that program was there from USA. Earlier we were begging food, so we wanted that our population will be fed properly. For the reason, we concentrated to breed the crop, the characters for which I can speak in between Hindi also, na?
Yes, yes, you can,
Yeah.
you can be bilingual.
Okay. तो तब की जो पॉपुलेशन था, आपकी पॉपुलेशन बढ़ चुका है, बहुत ज्यादा बढ़ चुका है, द प्रोडक्टिविटी इंक्रीज भी हो चुका है, लेकिन हम जो है वो प्रोपोर्शनेट इंक्रीज जो है, हम अभी इतना है कि हम अपने पॉपुलेशन को अच्छा से खाना खिला सकते हैं, एक्सपोर्ट भी कर रहे हैं, लेकिन क्या कॉस्ट है उसका? द कॉस्ट इज द केमिकल फर्टिलाइजर्स, द केमिकल्स compounds which you have sprayed, and we have deteriorated the soil too much without giving emphasis to that. And we bred the crop only with one program that will be increasing the yield. So overcoming the problem of hunger. Now, if you see, 8-9% of the world population live in hunger. So we want to feed them. To meet the food security need by 2050, keeping the population growth in mind, the food production must increase by 70%. So some of the crops are being given. This is Western calculations, but this holds true for us also. If you see rice and wheat and other crops what we eat in our country. Another thing, you go to any symposium and seminar, everybody says climate change, and we have to go for climate resilience agriculture. So these two terms, climate change and climate resilience, so these are going to be true in future, and they have profound effect in all aspect of agriculture. You cannot predict when the rain will come, you cannot predict when the drought will come, you cannot predict when the pest will come, when everything is now because of the climate change that is beyond our prediction. So we need crops which will be resilient to those conditions which we cannot predict. So climate change is also affecting the crop yields. So if you see maize and wheat, I didn't get in rice, maize and wheat. So China, Brazil, and France, those are global production is decreasing because of the climate change. Obvious, the traditional area where it is grown, in those area, whatever yield we are getting till now, because of the climate change, the proper photo period, proper growth is not there. For that reason, the yields are not there. This is simple thinking. There is no science involved in it. So now the crop growing area is being shifted. The traditional crop growing area is being shifted or will be shifted in future to different regions. The tropical region crops, which are earlier growing in tropical region, they will be migrating towards subtropical, and subtropical, the temperate region will be the temperature will be high. For the region, the subtropical crop, they will be migrate to the temperate region, like this. And where and definitely the situation is arriving, and it has arrived. There is no denying to that. So we have to future ready for food production and food security. And if you think about the different nutrient availability in comparison to what will happen in future. So the left hand side colors, left hand side red color, then after some little bit pink color is there. So those are the penalty. Means whatever we are getting from earlier crop, it is not that the crop yield is reducing, the nutrition quality of the crop is also reducing, because the plants are not able to uptake the nutritive element which they are earlier able to take into themselves, which comes to the food chain within us. So we are not able to get enough nutrition like protein, iron and zinc. So we are not able to assimilate those things from the crop plants. So now at this juncture, I'll ask a question whether you need food security or nutritional security. Food security is important to feed our population, and nutritional security is important to keep ourselves healthier. So Article 47 of the Constitution of India says it is a directive principle which directs the states to raise the level of nutrition and the standard of living and to improve public health as among its primary duties. And in particular, the state shall endeavor to bring about prohibition. Those are the other things. But our state directive principle says that we will be giving proper nutrition for proper health to all our citizens. So when you see about the nutritional security and food security, so yield, what are the different coordinates that determine yield? The plant architecture, high productivity, pre-harvest stress tolerance, reduced post-harvest losses. These are the four coordinates. Again, in nutrition, immunity boosting molecules, sometimes we call them secondary metabolites, which are important, essential amino acids, vitamins and essential elements. तो न्यूट्रिशन है क्या? न्यूट्रिशन यही है कि micronutrients required for the healthy human population. So for infant, adult and pregnancy, it has not determined how much nutrients and what are the different type of nutrients: thiamine, riboflavin, niacin, vitamin B6, folate, B12, iron, iodine, zinc, how much we need for our survival, for our good growth and for our good health. So as I told earlier, after the Green Revolution, the yield is increasing. But if you see the zinc content of rice plant, this is an example of rice. The zinc content in the rice plant is decreasing. So the penalty is the nutrition. We have increased the yield, productivity has increased, but again the same thing in case of iron. The content, the yield is increasing, but the content of iron is decreasing. So we have not given much emphasis to the nutrition of the crop plants when we start breeding for yield. So there are different desired crops which are required in the plant: high content of quality of protein, I just in a summary, I will not read all those things, but it is wheat, rice, maize, pulses, oil seeds, potato, vegetables, what we need actually. High content of quality of protein, high content of quality of oil, high vitamins, low toxic substances, and biofortified. And when you come the biofortified, there are three things. Through agronomically, we can increase the nutrients, biofortified the plant. Through in situ biofortification I'm talking. Otherwise, you can add any substance to when you harvest the crop, you can add any nutrient and substance and start blending them, and you call biofortification. But within the plant itself, so we can intervene agronomically to increase the nutrients. Through breeding, it is going since time immemorial. Through transgenic plants, the biotechnology comes into picture. So many people, as are told also, so there are बहुत सारे भ्रांतियां हैं. तो ट्रांसजेनिक प्लांट्स के थ्रू भी आप हम कर सकते हैं, न्यूट्रिएंट हाई न्यूट्रिशन रिच प्लांट्स भी बना सकते हैं. So there are 20 type of amino acids which makes up the human protein. We know that. There are essential amino acids. There are, we call them non-essential amino acids, but I will not call that non-essential amino acids. But all are essential, but more essential which we do not produce, we get from outside. So there are amino acid pathway we know. All the pathway we know. How those are being biosynthesized in the plant, or within us. Sometimes we know that, we know the pathway. So can you intervene in those pathway? Yes, we can. That biotechnology comes into picture. The status of different elements required by us. So there are different elements marked here in different color. So those are the elements essential for us because they are the cofactors of different enzymes. They carry out different reactions. Also, many times the chemical, the element itself act as the क्या बोलते हैं उसको, electron donor or electron acceptors to carry out different metabolic processes inside the body. So we require those elements. There are big 4 elements: carbon, oxygen. I can say that CHO, NPK, Ca, MgS, Fe, Mn, Zn, Co, Bo, MoCl. So these are the elements which you require inside our body. So these are all dietary nutrition, calcium, what I told, these compounds, how plants uptake them. The plants require them, they uptake them through different receptors, actively or passively. So there are different forms which take up these particular essential elements or different elements into the plant, and which ultimately through different means, through different transporters, it goes, and there are specific events and specific system inside the plants. I will tell that the genes, the proteins, they determine how these will be transported, uptaken, transported, and deposited in a specific place, not in all the place. Then only when they are deposited in the grain, that will be important. If they are deposited in the non-edible part, that may not be important for us, because we always go for the economical part which we eat. So similarly, the vitamins, the biosynthetic pathway of the vitamins we know. So all the biosynthetic pathway, can we intervene them? Yes, we can intervene, and we are intervening now. When I come to the genome editing, I will tell about that. So there are different pathways of, just I am giving a glance, no need to go inside the structures and complicated things. You can close your eyes at that period of time when you go for the, just I am brushing up and telling you what is crux of that. So there are different type of vitamins, and these vitamin pathways are known, how they are being biosynthesized. So there are genes involved in it, and we can intervene in those genes. So there are vitamin C also, citric acid and all other acids, those are being ascorbic acids. So this can be also manipulated, and people they go for breeding, either go for breeding or transgenic, and now genome editing they are going for to increase in different vitamins and amino acids inside the plants. Then if you take the example, biofortification of iron and zinc crops. So these are rice, wheat, pearl millet, lentil, and pomegranate. So this is actually the variety which are developed from ICR system. So 22.6% zinc in rice, wheat you can see 42% and 40%. These varieties are available. Pearl millet, these varieties are available. Lentil, completely iron content, high iron content. Pomegranate, iron content and very less zinc content. So accordingly, you can select a crop which are actually nutritionally important, and we require for our nutrition. Similarly, in all over the world, so there are different crops like rice, sorghum, maize, and wheat. So there are different crops of different amount of iron and zinc present in them. They are being developed, and we are in India. We are not less than that. So we are matching with the world nutritious crop which are which we have developed in our system, ICR system also. So enhancement of vitamin, protein, and amino acids content in different crops. I told about zinc and iron in that case. Now we have different crops which are rich in cassava, sweet potato, banana, and maize in provitamin A. We know the pathway of provitamin A. Then there are different lysine, amino acid, tryptophan, lysine rich, tryptophan rich, carotene rich, beta carotene rich, and we know the story about the golden rice also, how the genes from other sources they have been introduced into the rice plant to make the golden rice. So it is vitamin rich crops which are important, which are required for our health. The transgenic and genome editing, these two things I will touch upon. So genetic engineering of crop plants, why we need and what we should, what are the potential for disease resistance, insect resistance, biofuel production, herbicide tolerance, abiotic stress tolerance, nutrient use efficiency, then improving other things in biofarming, biofortification, metabolic engineering. I told about the series of biochemical pathway and genes which are available. We know all the sources, the genes are available. Sir also told about the synteny. There is not much variations between plant to plant in biosynthesizing these things because these are primary metabolic pathway and these are very much synteneous, means biochemical synteneous. So genes are available. So why not we can intervene in those things when we are nowadays intervening these things, and the next generation crop which will be coming out, those will be intervened crop which will be highly nutritious, high yield. Already we have done high yield, and now we are concentrating these high yield crops to increase the nutrition. Again, Sir told about the biodiversity. Now we are going back to our biodiversity and find out the wild type plants and what are their characters and disease resistance. Earlier, those were disease resistance, but now the crop plants are not disease resistance or insect resistance. Why? Because we have lost some of the characters during the time of breeding while increasing our yield. Now we are again going back to introducing those characters after studying the wild type plants into the crop plants which are giving actually high yield. Having said so, then there is a difference between traditional breeding, mutation breeding, and genome editing. So traditional breeding, spontaneous mutation that is 1,000 year or more it takes to fix it. Random mutation, 5 to 10 years. Then precise mutations, genome editings and all these things. This takes around 2 to 5 years. So traditional crop modification, around 10,000 years it started. Then genetic engineering, 1970, means when we started domestication, traditional crop modification they started. Then since 1970, and genome editing, it is a recent origin. So this is the strategies of plant domestication. And if you go to the ISA database, there are around 32 crops which are being intervened. I will not tell manipulation, genetic manipulation, intervened for transgenic production. And there are crops, there are genes, which are mainly a large number of genes are Bt toxin genes, Bt not toxin, Bt protein gene, which is used for insect resistance. Also, some of them are converting some of the fatty acid to other type of fatty acids. But because many a times industrially what you need, the oil should be tolerant to high amount of temperature, pressure, and humidity in the food processing industry. So, but the essential the oil which we eat, what do we want? They should be rich in uh, unsaturated fatty acids. So there is a compromise between these two. If the saturation is increased, then the oil will be more tolerant to high heat and humidity and all industrial processes. So palm oil, which is being imported, maximum food processing industries they use the palm oil because of the palmitic acid. This is saturated acid. But for our development, growth and development, and for our health, we need unsaturated, that one omega fatty acids. Hence, the people they started playing with the fatty acid biosynthesis gene and started producing Brassica napus with different composition of fatty acids, which can be used industrially as well as used domestically and other uses. Similarly, the starch manipulation, amino acid increase in the amino acids. So in this database, around 32 crops are there. I have listed them. You can go to the CSIR database and find it out. So GM crops approved in India. So these are mainly the in cotton, and one is glycine max, BASF. This is not approved. This is actually we are importing as the poultry feed, for poultry feed. After the government of India pressurized, because glycine marks was not available. Again, what the poultry feed industry they need the uh, the feed which is rich in amino acids like D and L, methionine, arginine, all these amino acids are needed. And in our country, we are not producing those amino acids as such. And whatever amino acids are being produced, these are from the synthetic sources, petroleum sources. When we use those, we cannot say that organically grown chicken. Now, organically is a buzzword. We should grow everything organically. For that reason, to get high the glycine max, that is soybean, rich in high content of amino acids, so we are importing the transgenic plants, and we are telling that these we are giving organic food to the organically grown chickens. Like this, the Gossypium hirsutum, that is cotton, that is approved in India. So these are the two crops which are approved in India. And till 2022, again I have noted down which are the crops and which are the plants. Those all are available in ISA database, and these are in different countries which are available. And coming to the genome editing. Also, sir told about the CRISPR-Cas. I need not go about the technology. The thing is that there are three types of genome editing: SDN1, SDN2, and SDN3. SDN1, we can delete in one nucleotide or add one nucleotide, create a point mutation. SDN2 is we can go for modification of some of the nucleotides or one nucleotide. SDN3 is just like transgenic plant. You can take a gene outside and put it inside the plant. So SDN1 and SDN2, because they don't need any gene integrated inside the genome from outside, for the reason they are being approved by government of India. Then ultimately, now all of us, we are going for the genome editing. Here, the institutional biosafety committee will be responsible. I was in RCGM when it was being passed. The same day we have conducted the meeting, all of us, and it has been passed in the same day because there was a pressure that we have to go quickly for approving the SDN1 and SDN2. And the protocols and the SOPs are now in place. Now, all of us, we are going for genome editing in large scale, but question is the process of genome editing. This is patented from two places. So we cannot circumvent this patent, and we have to either purchase the patent, because this process is not of our own. Whatever SDN1 or SDN2 material we will produce in crop plant, we cannot grow it commercially. Even if for research purpose, we need a research license. So for that reason, now Government of India, there is a interministerial group in which I am also a member. We are discussing how we will be actually getting those patents, or we will be doing some research such a way that we can have our own patent in carrying out those SDN1 and SDN2 genome editing in India. So that is really a problem. So we are facing this problem, and we will find out a way, and in future we will be coming out with a solution. So these are the different success story CRISPR-Cas. I told we create mutations just like normal mutations through CRISPR and Cas. cash these processes. I'm not going to tell it here. So from 1912, we have blight resistance rice. 2015, powdery mildew resistance wheat and herbicide resistance canola, they came. Then high yield waxy corn, soybean with reduced trans fat, non-browning mushrooms, polyphenol, intervening in the polyphenol oxidase pathway. Then potatoes with altered starch, virus resistance cucumber, high yield rice, gluten-free wheat, domestication of wild tomato. So if you intervene in 3 or 4 gene, the wild tomato will be domesticated tomato. The non-gluten, non-transgenic wheat, development of cyanide-free cassava, the intervening in the cyanide biosynthesis pathway. There is GABA tomato. This tomato, only intervening in the single gene. Then there are D3. We know that cholecalciferol, vitamin D3. This pathway exists in different plants. In some of the plants, the pathway operates in a low level. In some of the plants, it operates in a higher level, like the plant like Cestrum diurnum. So in tomato, there is a single step intervention for production of cholecalciferol. So a serving of gene edited tomato has as vitamin D3 as 2x or 28 gram of tuna piece. So now we, the plant scientists, are coming out with, with such crops which are novel and which can provide alternatives for our health benefit and nutrition in addition to maintaining the yield. Then genome editing for vehicular iron. That's an interesting thing. Mechanism it not being known, but what they did, they created some of the mutation, CRISPR-Cas mutation, and one of the mutations they found instead of leaf sheath, the iron is deposited in the embryo. So that is good for us because we need the rice grain we eat. So if it is deposited in the embryo, then it will be good instead of deposited in the leaf sheath. So these are the type of work going on in all over the world. Now target gene being used for genome editing CRISPR-Cas in India, that is the ICR work. So if you see the different genes which are being taken up by ICR, then you will see the right hand side column, the country of origin of those genes, either they are being published or patented. So not a single gene is identified from India. Always you see China, USA, Japan, South Korea, France, Denmark. I have not found out a single gene from India, but we are using those for doing genome editing in our own crop for improvement. Whether we will face the patent problem or IPR right problem in future or not, I don't know. Individually, we have to go for negotiation with those groups who has the group and who has the gene and the patented. So what should be the focus? Genome medicine, improved crop yield, disease resistance, improved quality, crop quality, herbicide tolerance. Then there are different hybrid breeding, cold, drought, and salt tolerance, grain yield, grain size, oleic acid, carotene. These are all the nutrients, fragrance, amylose, anthocyanin, cadmium, storability, glyphosate. Then all other things, different herbicide tolerance. Then thermosensitive, genetic male sterility, everything which are actually integrated inside the plants can be induced through genome editing, just like the wild type, matching the wild type and matching the high yield of the future, what we need. So those can be actually induced inside the plants to produce future super crop to meet the demand of higher population which are growing. We cannot stop the population. We cannot reverse the climate change. We have to grow crop and we have to produce crop according to the need in future, which we are doing right now all over the world. I'll not go for this C4 cycle, but can we afford to ignore the technological advances in agriculture? Previously, this was the agriculture, but we got tractor. Now we have adopted this thing. Like this, the transgenic plant, these are being supported by all these organizations which are actually the need of the power. We in future, when the population will be increasing so much, and we are ready with many transgenic plants in all over the world, including in our country, which will help to increase the yield and nutrition of the plant, we will definitely adapt it instead of dying. We'll eat it definitely in future, and the time is not far, which will be coming up quickly. Genetic enhancement of yield of nutrition, example of rice. So this is brown rice, wheat, maize, and potato. So I have made a comparison how the rice nowadays, we are very crazy about the brown rice. What is the nutrition? And I have also written down some of the intervention at amino acid level. What I have told, just I have summarizing this thing: enhancement of lysine content, enhancement of theorin content, enhancement of tryptophan content. All these things we can, and we are doing, and we will be doing in future for the betterment. Genetic engineering interventions. So there are many pathways and all these things which are taking over, which are actually going on all over the world, and different crop like rice. There are different genes which are being from either from homologous source or heterologous source, from other from bacteria also sometimes. So the QTLs, coming to the QTLs, only generating QTLs and integrating QCL and finding out the linkage drug or enhancement, that is not enough. We have to enter into the QTLs, and what are the gene responsible for the QTLs. We have to describe them functionally, and once we describe them, then we can develop marker to screen the plants in future. And many of the rice plants and all the other plants, people are working on QTLs, and they are developing markers, and marker assisted breeding is helping nowadays. And the new generation crops which are coming up right now, so these are all marker assisted crops, they are coming up. And and many crops which will be coming up in future also, those will be marker assisted in long run. After knowing the mechanistic history of the genes present in the QTL, what they are doing and how they are working, and what are the different characters they are regulating. The genome editing rice all over the world. So this is the status. So again, the genes specifically I have written down. Then coming to the plant immunity, uh, I told about the a whole scenario of agriculture and how actually trying to improve it. But my personal belief is that plant has got its own immunity. So instead of taking the genes from outside and inside and making it transgenic, why not we induce the gene from inside the plants, which are being defective since we have started improving the plants from domesticating the plant and improving the plant. So plant immunity, this is the टीडियों के प्रकोप से बचे औषधीय सघन फसलें. सीमा में एक लास्ट टू लास्ट ईयर टीडियों का अटैक हुआ था जब कोरोना टाइम पे. तो सारे क्रॉप प्लांट जो उसमें ग्रो कर रहे थे सारे मर गए, लेकिन मेडिसिनल एरोमेटिक प्लांट ग्रो कर रहा था. वो खाए नहीं, चले गए. मेडिसिनल आर्टिमिसिया, लेमन ग्रास, उनको कुछ छुए ही नहीं, बाकी सब साफ कर दी. क्यों? क्योंकि उसमें तो कुछ तत्व है. Something is there in the medicinal aromatic plants. So these are very much important. So in CMAP, as Sir told also, we have sequenced the whole genome sequence of the CM Ayu, that is the Tulsi, Osimum tenuflorum, and we have gone for different comparison of different species with the biosynthetic pathway and novel molecules. And this is the sequence. What it revealed? It revealed many novel flavonoids, and we could identify many pathways, flavonoid pathways. Then strategic genome resources we have now in Osimum, including Osimum mentha and different Osimum species, Symbopogon flexuosus, Pelargonium, that is, and Chrysopopum ginger nuts, that is vetiver. We have the genomic resources, and we have identified many monoterpenes biosynthetic genes. And these genes can be utilized in future for synthetic biology, and we are utilizing this gene. As for example, these are the sesquiterpene genes, and we have gone for introducing and improving viridiflora in mentha, where when previously it was producing around 0.5%, in the essential oil. Now it is more than 25%. So the fractionation will be easy. And this is a high value molecule. They have been costing around 10 milligram around $300. And this can be used as an artificial hormone. Also, in the perfumery industry, high value perfumery industry, to in, to increase the lactation of animal also, this has been reported to be used. Then Centennies are told, Centenny I told, I am referring, how much time I have, Monica?
Sir, we can go for 10 more minutes.
Okay, otherwise I'll go on. So Centenny as I told, I'm not telling about the genetic Centenny as such, I'm telling about the biochemical synthetic, enzymatic Centenny. So sesquiterpene is found in many plants, but some plants are dominant. The biosynthetic pathway and the genes are present in all the plants, but somewhere the expression is dominant. Sesquiterpene, monoterpenes, diterpene, tetraterpene, it is like that. So there are many such compounds which are related compounds, and the pathways are present in many plants. So, and many insecticidal compounds are found in those secondary metabolites. And for that reason, if I ask a question to myself, why some of the insects, they prefer rice plant, not wheat plant, the question lies in that the secondary metabolites. And I actually interrelates those things to the immunity of the plants, the secondary metabolites, how they ward away the bacteria, fungi, and biotic stresses like insect and pest. So this is one example. If there is increase in cis-hexanal, then there will be virus tolerance, because it somehow induces the callose formation, and for that reason tomato leaf curl virus resistance will be there in tomato plant. So the secondary metabolites induces some of the mechanism by which the plant will be resistant to either virus, fungi, bacteria. I will come to the second example. This is Withania somnifera. Though this plant is uh, some of the Withaferin, they are responsible for fungal stresses, tolerance to fungal stresses. Another example, this is from our work in potato. So farnesyl actually when you induce the farnesyl biosynthesis in potato, then late blight tolerance is increased. Then in pod borer, this is NIPB's work when we're working in Cajanus platycarpus. Sir told that we have to learn from the wild sources. So we have sequenced the whole genome of Cajanus platycarpus, and we found out some of the genes which are present in Cajanus platycarpus producing secondary metabolites, epicatechin 3 compounds. They are actually conferring resistance for this larva, Spodoptera litura and Spilactera obliqua. Other examples, this is the genome sequences in NIPB. I was in NIPB, so I'll tell about the NIPB. These are the genome sequences which I come from there. So these are the variety through marker-assisted breeding which have come up from rice, mustard, wheat, chickpea, and pigeon pea in NIPB in collaboration with other institutes. So through biotechnological means, marker-assisted breeding, these have come up. And there is a Cajanus cajan, that is arhar plant, the arhar dal plant, that is being resistant to Helicoporba armigera, the pod borer. So now these plants is in the advanced stage of trial. So it can be NIPB will apply for future release of this plant. Again, Sir was telling about NBRI's work, the tomato ripening. The genes can be ripening can be stopped or not stopped. It can be delayed by different genes. So this can be either done through RNAi or genome editing. Again, the molecular basis of petal abscission in different petals. We want the flowers to stay for a long time in our home. They should not go bad within 1 or 2 days. For that reason, the ethylene biosynthesis you can stop, then petal abscission can be stopped. So this work is being done in NBRI. Then root growth and drought tolerance. This work also is being going on at NBRI. So we have around more than 200 pangenome already being done, and we will be developing a chip in recent future to analyze the background and foreground of all the plants and develop the cotton genotypes in future. Also, you can test which part of the country from outside the cotton has come entered into the India to produce the textile. This is in mango. This is a climatic fruit like tomato. So how they are ripening and what are the different molecular basis of ripening, molecular basis how they are ripening, as well as how you can delay the ripening, along with what are the molecules present there, and these molecules can be taken up for developing different aroma, which can maybe in 3-4 years we will get those aroma, natural aroma compounds synthesized in vitro, synthesized, semi-synthesized in yeast system or bacterial system, and you will be eating ice cream. Then the rice, the arsenic rice, Dr. Devasis is working in NBRI. So he has developed a Muktasri along with in collaboration with uh, West Bengal, one of the institute in West Bengal, uh, Chinsura, that is RSS Chinsura. So this variety is a mutant variety which actually uptake less arsenic. In addition to that, he has some of the receptors. If we go for inducing the mutation in those receptors, so you will get less arsenic in the grain of the rice. We know arsenic is a problem all over India now. It started from Bengal and it is now invading everywhere near the Bengal, Bihar, and now it has come to Uttar Pradesh also. The reduced arsenic accumulation in seeds variety 50% via volatilization. One of the genes we actually volatilize, methyl transferase gene, make 3 methyl group. So these genes are being identified and it has been introduced. And another example of that is Marchantia. The gene from Marchantia can be introduced into the rice for less arsenic uptake or less arsenic deposition. And some of the transporters, they will not deposit in the grain. Those are being found out that inositol receptors like thing, those genes were now being genome edited, and they are being found out that it is not deposited in the grain, though it has been uptaken from the root. So what are the approach now? Genome editing, allele mining, identification of genes, mainly those are master regulators by functionally validating them in plant, mechanistically analyzing them, overexpression of heterologous gene in a stress-inducible tissue-specific manner under a marker-free environment, a robust transformation protocol, all vectors, cassette, and tools for genome editing to be developed indigenously, and finally the genome editing and identify master. I already I have told about these things. Way forward is genetic engineering and gene editing are powerful tools. I have told. Then GM technology will play a crucial role in empowering farmers, feeding global population and protecting environment in future. Need for indigenous IPR protected target repository for genome editing. All the genes now onwards, we have to find out our own genes, analyze our own genes, functionally analyze them, and get IPR for them. And we should have a repository of Indian genes now. Need for long-term climate resilience research, because the short-term stress research is gone are the days. We can have short-term stress research, but in future, which type of stresses we will be getting when and how long, we don't know. So we have to have crop plants, robust crop plants, which are resilient to climate change in future. So with this, I will stop, and once again, I will bow my head before my teacher, as well as all of you, my friends, colleagues, and whoever is listening. And thank you very much for giving me this opportunity. Thank you. Namaskar.
Thank you so much, sir. So just
I don't know whether I have done the justice or not, but if anything left behind, Sir can speak.
thank you so much, sir, for the thought invigorating talk. We walked through climate change scenario and production values, also the relation of yield and nutrition through time and biotech interventions, CRISPR-Cas, genome editing, biofortification, also to plant immunity with special reference to maps, as you said, pollutant uptake solutions through genetic engineering, and much more. Thank you so much, sir. It was a really wonderful talk. Sir, if you allow me for a question, we have a quick question and then we can
Now, if I can answer, then you can ask me.
Though I would like to congratulate Professor Shasany for very excellent presentation and very informative knowledge about the application of biotechnology for the improvement of crop. Thank you very much. It's a wonderful lecture, and it was wonderful moment for me to listen and continue to listen your lecture. Thank you very much, and congratulations for doing a very excellent lecture. At this moment, I would like to appreciate you for your kindness, for your expressing your kindness about your teacher, Professor Khanuja, and for his and for expressing your acknowledgement for your work and for your scientific career. That's a very, 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 very expressive and very पाथ वेरी एक मॉडल है और यंग जनरेशन के लिए कि आपको अपने टीचर और उनके लिए किस तरह से आपको अपनी फीलिंग्स रखना चाहिए और टीचर्स को किस तरह से रिजल्ट थैंक यू वेरी मच फॉर योर एक्सप्रेशंस और एक्सप्रेसिंग योर acknowledgement and for your work for your teacher Professor Kanuja. Thank you very much.
Thank you, sir. Thank you. Thank you.
So I have a request from Founder President. So if you allow us, we can take that question and then we can
Yes.
open the session for internal discussions.
Yes.
Okay, okay. So we have a question on Facebook itself. Anuradha Mahi says, very informative talk, sir. Thank you. One question though, you talked about insect resistant gene. Is there any ecological consequence to it?
Yes. When we go for genes from outside or any other sources, then as such, if it is Bacillus thuringiensis or something like that, insect resistance, I don't think any ecological consequences are there because it is present already in the environment, and how it is going to have some effect to ecology. So there is I don't understand whether it will be having or not. This is in our brain that we will be having some ecological consequences, but there will be none. I can say that with full confidence. The second thing, when you are going for insectical genes from the plant by itself, some other plant, already it is there. The genes are already there in the ecology. So you are only taking from one plant to another plant. So I don't think any ecological imbalance will be there in that case. This is my personal thinking.
Thank you, sir, for answering the queries. So, Kanuja, sir, please, I request you to take the stage.
Extending into what Dr. Shasany said about the ecological consequences when you use genes from nature. Well, nature itself evolves. So those of the environmentalists who feel that nature doesn't evolve are wrong. And evolution means genetic changes only. All evolution is consequence of genetic changes and selection that is happening in nature. And he very rightly said that if you take example like Bacillus thuringiensis, which itself is present in soil and also present on phyllosphere of plants and produces that toxin gene called crystal protein or delta endotoxin. So it is not toxic to human beings. The receptor, if you look for in the human gut, is different, which doesn't really allow it to be the toxin, while insect gut has those glycoprotein receptors on their brush border membrane vesicles, and that's why they get eliminated. So basic thumb rule, which I would like to say is, if you go pro-evolution, there is no harm in genetic engineering or gene manipulation. If you go anti-evolution, then there can be consequences. So that's the caution that people will have to take. Uh, Thank you, Ajit, for giving a wonderful talk. It really translated the way I felt it should, and has given both the students and the society the knowledge about biotechnology, how it is being used and how it is going to be useful and how it is going to be essential in future. Without this, we are not going to be able to survive even for our food, leave aside nutrition. And you very rightly linked food and nutrition. So I would say in today's silver talk, the silver lining that Ajit has highlighted consists of that as we kept on increasing the yields, that means the food increased, but nutrition went down. Now, that is quantitative and qualitative. These are the two aspects of any crop. So if you don't take care of quality along with quantity, you're going to end up into such consequences. Green revolution was need of the day. It really served the purpose. And learning from green revolution, how we can make it evergreen revolution is what biotechnology is going to do. Second silver point which she has made today, which I think particularly students and researchers have to follow up for future research, is that wild resources, wild genetic resources are the key. It is said that the successful genes of today are the defeated genes of yesterday. So don't discard the knowledge. And that's why when we are going into genome sequencing and structural and functional part of the genomes, we must keep that into our library, that tomorrow with climate change, anything happens, you will get those. And those are called adaptive functions. You may go for yields, you may go for increase in productivity, but you may not be able to adapt to the change in climate that is happening so suddenly now. And it is consequence of what we have been doing with nature. And therefore, for those adaptive functions, we will have to go into wild resources, which are still remaining intact in spite of such climatic shocks that are happening. And that's where we will get those genes. And third was, me and Dr. Shasany both have a linkage with that, and that is medicinal and aromatic plants. That medicinal and aromatic plants are not only aromas and drug molecules, but they tell you a very different scenario of physiology and metabolism or biochemistry through the genomic constitution, such that what he referred as immunity is that जो हम हिंदी में कह सकते हैं कि रोगों के प्रतिरोध और कीटों के प्रतिरोध की जो क्षमता है. So the immunity or resistance or tolerance or resilience to abiotic and biotic stresses is what medicinal and aromatic plants are going to serve as the models. So I think that's what I would like to summarize what Ajit said, and I'll pass on to Dr. A.K. Singh for further deliberations.
Thank you, Dr. It was a pleasure to hear Dr. Shasany after a long time. एक बड़ा सुखद अनुभव रहा है और ज्ञानवर्धक अनुभव रहा है, न केवल हमारे लिए बल्कि जितने भी श्रोता थे, किसान, विद्यार्थी, जैसा कि आपने बताया. और डॉक्टर शासनी इज अ वेरी हार्ड वर्किंग साइंटिस्ट, वेरी innovative scientist, with innovative ideas, and he's a hard taskmaster also, as Professor Sanuja. So I think it will give a lot of insight to the young students और किसानों को भी ये सोचने के लिए कि भविष्य के गर्भ में बायोटेक्नोलॉजी से बहुत ही उपयोगी चीजें छुपी हुई है. मैं जब भी कभी गांव जाता हूं तो किसान कहते हैं, जैसा कि आपने बताया, कि भाई इसकी उपज तो बढ़ गई, लेकिन खाने में वो आनंद नहीं आता है, स्वाद नहीं. शायद आपने बताया कि उसमें न्यूट्रिशन भी गिर रहा है. तो भविष्य में जो आने वाली सुपर क्रॉप्स हैं, जो फसलें हैं, I think these will solve the problem. And जो आपने बताया कि किस प्रकार से बायोटेक्नोलॉजी का जो रूपांतरण, ट्रांसलेशन होने जा रहा है प्रयोगशाला से खेत की तरफ, इस पर भी ध्यान दिया जाएगा और इसमें डॉक्टर शासनी का बहुत बड़ा योगदान होगा. हम लोग कई वर्षों से, बल्कि पिछले 1-2 वर्षों से इस मंच पर उन्हें लाने के लिए प्रयास कर रहे थे, लेकिन वो एक मिशन में थे. और अच्छी बात है कि वो लखनऊ में है और भविष्य में भी हम इनको कई बार कष्ट देंगे और जो नए जो अनुसंधान होंगे, जो उपलब्धियां आएंगी बायोटेक्नोलॉजी के क्षेत्र में, वो हमें बताते रहेंगे। बहुत बहुत धन्यवाद डॉक्टर शासनी फॉर स्पेयरिंग योर वैल्यूबल टाइम फॉर गिविंग दिस वेरी इंफॉर्मेटिव लेक्चर। साथियों, ये जो वेब सीरीज है, इसमें हम अगले माह सेकेंड सैटरडे को एक नए एक्सपर्ट के साथ, एक नए विषय के साथ फिर उपस्थित होंगे। तब तक के लिए नमस्कार, धन्यवाद।
So if you permit me, can I say
Sure.
actually next week, starting from Monday to Saturday, NBRI is celebrating its 1 week 1 lab program. So there will be 5 theme area for 5 days, and all days there will be a theme area for farmers in the first day. Second day there will be theme area for environment, and in August 14th, 15th, we will be having theme area for plantation and beautification of Lucknow.
Okay.
And next biotechnology as well as product development. So these are the area. So there will be exhibitions, stalls, and panel discussions. So I will invite everyone to please come, and all the students mainly, the students, they please come and learn and how the things are going on and how the research is going on into future generations. Thank you, sir.
Thank you very much.
Thank you for this information, and I think most of the students they will make use of it, and we shall also be trying to participate and see.
Ajit, if you also tell the timings for all the days, when that will be open.
So I will be sharing the program schedule in Florafana. So so it starts from 10 o'clock every day, ends at 4:15 or 4:30 every day.
तो 10 बजे से 4 बजे तक लोग वहां जा सकते हैं।
हाँ, there will be experts in the field. They can listen to the experts, how they are, what they are speaking, and which direction the research is going on. So, and also the exhibitions, different day, different type of exhibition according to the theme area.
Very good.
Very good.
Thank you. Thank you, sir.
Okay, sir. Thank you.
Thank you so much, sir.
Thank you, sir. Thank you. Again, sir, happy birthday, advance happy birthday.
Thank you. I'm growing old, that means.
All of you. From all of you.